question is that I don't think anybody has studied the epigenetic implications yet, at least in any for another base pair. But the irony is that the standard genome editing methods, including the use of CRISPR-Cas9 nuclease, have difficulty correcting these single nucleotide polymorphisms.
And because of that, we can now start to collect many individuals' genomic information.
Well, we know that technology is very powerful. It's basically a molecular scissors. We can cut DNA and make changes, but it's unclear how precise that cutting is. And so the worry is that you cut in places you don't want to cut, inactivating genes that are very important, or maybe changing the expression of genes that could predispose to other problems like cancer. So we're clearly still getting a grasp on the science. And then, of course, there are the broader, deeper societal questions of what kinds of conditions might this be used for. There's certainly more acceptance of the use for alleviating disease, alleviating suffering. But what about just disease risks? And what about traits that we might call enhancements? Should this be a technology that we use to affect a child's ultimate height, their skin color, maybe their various other traits? That on which are the basis of much of our discussions.
I think, especially because the technology is moving so fast, we really need to have open dialogues such as this summit here, as well as transparency in the way that people are carrying out their work. And I think as we push the technology forward, we already do see scientists and also medical professionals sort of collaborating together to really put all their brains together to think about the best way and most responsible way to move it forward. And I think we need to encourage more of that and to make sure that we're really doing the best we can to get therapies developed responsibly and also quickly to get to the patients.
Greater investigation, but there is enough of a diversity of cytosine deaminases that have been